Can DNA be extracted from ancient bones? Have you ever wondered if we can actually retrieve DNA from ancient bones? This fascinating question opens up a world of possibilities in archaeology. Yes, DNA can indeed be extracted from ancient bones, and this process plays a vital role in archaeological osteology. This field focuses on analyzing human remains to gain a better understanding of past populations. The extraction of ancient DNA, often referred to as ADNA, allows researchers to study genetic information from individuals who lived hundreds to thousands of years ago. The process begins with careful sampling of bone material. Researchers typically target dense parts of the bone, such as the petrous portion of the temporal bone or teeth. These areas tend to preserve DNA better than other skeletal elements. Once the bone material is collected, it is done under sterile conditions to avoid contamination. The next step involves chemical treatments to isolate the DNA molecules. There are several extraction methods available. For instance, carrier-mediated ethanol precipitation and isopropanol-based techniques have proven effective. These methods help recover DNA from ancient bones while minimizing contamination risks. The preservation of DNA in ancient bones is influenced by environmental factors. Temperature, soil chemistry, and burial conditions all play a role. Bones exposed to high heat, specifically above 200 degrees Celsius, show significant DNA degradation. However, bones subjected to moderate heat, such as those affected by fires up to about 350 degrees Celsius, can still yield usable DNA with specialized protocols. Once the DNA is extracted, it can be amplified using polymerase chain reaction techniques. This process generates enough material for genetic analysis. Archaeologists can study genetic relationships, population movements, disease history, and even aspects of evolutionary biology through this analysis. In archaeological contexts, the extraction of DNA from bones is conducted in dedicated clean laboratories. These labs are equipped with ultraviolet light and filtered airflow to minimize contamination. The protocols are highly controlled, often involving multiple steps of decontamination and careful sampling from specific bone regions. This meticulous approach maximizes the yield of DNA. Overall, the extraction of DNA from ancient bones is a well-established practice in archaeology. It provides genetic data that complements traditional osteological analysis. This combination helps reconstruct the biological and cultural history of past human populations, revealing stories that have been buried for centuries. So, the next time you think about ancient bones, remember the incredible potential they hold for understanding our shared human past.